Hello everyone, welcome to Medics 2023. My name is Shobhna Kapoor and I'm an Associate Professor of Chemistry at IIT Bombay. We are a team of chemists and biologists which mainly work in developing new tools, technologies and assays for both therapeutic and diagnostic utility in tackling infectious diseases. And today we are here to pitch our platform which may help in expedited discovery of resistance-free membrane-active antibiotics in tuberculosis. Now, in tuberculosis, historically there have been a number of unmet clinical needs as displayed here, which is further confounded by the emergence of multi-drug resistance. This issue of deadly resistance standalone is enough to bring out the limitations of the current solutions that exist for treating tuberculosis, which is namely the protein targeting drugs, which due to their long treatment times and high doses generate resistance. This collectively shouts out the need for other molecular targets for treating tuberculosis, but luckily we do not have to look very far because one of the most commonly used resistance-free approach in treating fungal infections is commercially available, known as Ambizome. It is a small molecule that targets fungal membranes selectively. So thus, if we want to have similar kind of lead in treating tuberculosis, we need to follow a few steps. And the place where our technology comes in is where we are able to develop membrane platforms for screening for similar kind of compounds in tuberculosis, which can be expedited, targeted, and selective. So what is our technology? It is molecularly complete cell-free membrane scaffolds derived from the cell envelope layer of mycobacteria as shown here. So it is physiologically relevant in terms of its model composition. Other salient features of this membrane systems are that it is protein-free, so therefore it helps you to screen for molecules that do not have any protein mode of action. Another advantage is that they can be designed in various geometries such as liposomes or solid supported bilayer configurations, suitable and adaptable to different high throughput screening. Once you have these membrane scaffolds which are fully characterized and contamination free, you can use them using absorbance and fluorescence based high throughput screening to identify for chemical compounds that can disrupt bacterial membrane lipid bilayers, go on to confirming whether they are able to show the similar effect in killing the bacteria, and identify only those compounds which have a membrane mode of action, hence resistance free, and not targeting proteins because you are starting with a system which does not have proteins to begin with. So all in all, it actually helps in making the transitioning from hit to lead pipeline quicker. In terms of market statistics, India along with Southeast Asian countries covers or represents the majority of the PAPIN population, only one step behind South Africa. And as mentioned, currently there exists no treatment in terms of resistance-free approaches for treating tuberculosis. And we think that our membrane technology might provide an avenue for faster treatment of tuberculosis while obviating the resistance issues that have emerged over many years, eventually improving the quality of life of our people. Thank you, and I will be happy to take any questions.